Namaskar. Welcome to our channel Bio by Gulab Sir by Wisdom Coaching Center. As a series of lecture, today we are continue to learn in search of heredity material under the topic molecular basic of inheritance. There are three group of scientists that they had discovered that DNA is the genetic material. But before that there was misconception about hereditary material, that protein would be the genetic material. It was said that protein would be the genetic material. Why? Since it is ubiquitous, found everywhere, and therefore it was believed that protein would be the genetic material. But by these three group of scientists in a series of experiment, that three series of experiment, one, transformation principle, two, molecular explanation or biochemical explanation for the transformation principle or Griffith experiment, and third, Hershey and Chase. These three group of scientists with three series of experiment, they could prove that DNA is the genetic material but not protein. As a part of these three experiment, we are going to learn today first experiment given by Frederick Griffith, principle of transformation under molecular basic of inheritance. So, in search of genetic material, topic is in search of genetic material. Under this heading, principle of transformation, principle of transformation. Transformation. This experiment given by Frederick Griffith in 1928. In 1928, Frederick Griffith has given an experiment. He selected a pathogen, streptococcus pneumonae, selected pathogen streptococcus pneumonae for his experiment. Streptococcus pneumonae for his experiment. These streptococcus pneumonae are of two types, that virulent and non-virulent. Virulent, that is S-shaped bacteria, S-shaped bacteria, and non-virulent are R-shaped bacteria. This S-shaped bacteria, because of smooth covering around bacteria. This was the smooth covering around that bacteria. So, smooth covering that protect it from antibody of that host. This is a pathogen. When it got disease, it get enter into the body. This get protected because of this mucopolysaccharide smooth covering in smooth S-shaped bacteria. R-shaped bacteria, they do not have this covering, and therefore they do not cause disease. So they are disease causing. Virulent means disease causing, and they are non-disease causing. So he selected these two strains for his experiment, and what he has done, he has injected. So he carried out his experiment in 4 steps. That first step, that he injected this S-shaped bacteria. He injected S-shaped bacteria, S-shaped bacteria, injected into mice body. Mice died. As a part of series of his experiment, steps of his experiment, he injected R-shaped bacteria into mice body, survived. See, when he injected S-shape, disease causing, caused pneumonia, mice died. He injected R-shape, non-disease causing, do not got pneumonia, mice survive. But when he got success to kill that bacteria by heating. So he injected, killed H. bacteria. Heat killed H. bacteria. H. bacteria injected into mice. Mice survive. Means heat killed bacteria could not get or could not develop pneumonia in mice survive. But as is again step of his experiment, he injected heat killed S-shape plus R-shape bacteria injected into mice. Mice died. He got surprised that heat killed S-shape do not develop the disease, R-shape injected do not develop the disease. But why heat killed S-shape along with R-shape? When injected into the mice died, and from dead body, very important, and from dead body he could extracted only S-shape bacteria. Then he surprised that where R-shape bacteria has gone, because R-shape bacteria were injected, S-shape bacteria were not injected, it was heat killed. Then what happened with the S-shape? What was that left behind in heat killed S-shape bacteria that could transform this R-shape bacteria into S-shape? Then he concluded that might be the material, that might be the chemical, that might be the substance that transform R-shape into S-shape. So, although he could not give biochemical explanation for that genetic material, whether it is DNA, RNA, protein, but he just said that would be the material which left inside the heat killed S-shape bacteria that could transform R-shape into S-shape. So that is the transformation principle. To conclude, something that left in heat killed S-shape bacteria that is a transformation principle in heat killed S-shape bacteria, transformation principle to transform R-shape bacteria into S-shape bacteria. So decided that would be the genetic material. So he proved that would be the genetic material which could responsible to transform R-shape bacteria into S-shape bacteria. And now that became a base for the next group, for the next experiment given by second group of scientists, MacLeod, McCarty and Avery. So here they could explain that genetic material is nothing but it is the DNA that transform R-shape into S-shape. But who? Second group of scientists. Griffith could not. Griffith only said that something left inside the heat killed S-shape, that is the transformation principle, and that would be the genetic material which could transform R-shape into S-shape. Up to this only Griffith completed his experiment, and that became a base for the experiment given by second group of scientists, that is biochemical explanation for this transformation principle given by MacLeod, McCarty and Avery. And third experiment, that Hershey and Chase. So in second lecture that we will learn about biochemical explanation for transformation principle given by that three other group of scientists. So as a part of series of that experiment, to prove that DNA is the genetic material. That first experiment given by Frederick Griffith in 1928 as a principle of transformation is what we learn today. I hope you could have understand, you did understand, and question can be asked in board that for 3 marks, what is transformation principle? Explain it in detail. So these are the steps, these are the key words and key sentences are sufficient enough to get complete 3 marks in that question. Even if not explanation given, as it is also written, it is sufficient to get 3 marks. Moreover, in NEET, there may be many questions from this topic. So as a part of this topic, conceptual learning is completed. Therefore, don't worry, you will be able to understand the question and apply your knowledge to solve any question asked on the basis of Griffith experiment. So be confident, enjoy by joining series of our lectures. I hope you did understand still. If you are in doubt, you can ask in comment section. I appeal you to like our channel by subscribing it, and by pressing bell icon, you will be notified as and when new videos will be uploaded. Thank you.